Uh, this is Koji from Duke University. I'm a member of the Early Investigator Committee of ISBM, and we organize a monthly webinar seminar. And it's my pleasure to moderate today's lecture and with Dr. Lachaner, who are the postdoc of Washington University. And before we begin the seminar, I'd like to make a brief announcement regarding to the ISBM 2026. So International Society of Bone Morphometry is an international society focusing on bone imaging. And one of the aspects I truly appreciate about this society is it's very highly interactive and it's welcoming and inclusive community. And the next ISBMR meeting will be held in London, UK. Of course, beautiful place. I never went to there. And abstract submission for this meeting is currently open. So I highly encourage everyone to register this meeting if you follow the bone or muscle and something image. And now it's my pleasure to introduce today's webinar. But like I also encourage to register the LinkedIn follow to catch up the monthly webinar. Because like we will have a busy month for next month. We will have a lecture from like Professor Protkin and the Professor Hunter about how to writing an amazing abstract, and which will be held for February 3rd. And also we will have our monthly webinar on February 26th. Okay. Now I'm going to introduce today's lecture. Professor Frank Core and Dr. Core received his training bioengineering at Rice University and obtained his PhD in mechanical engineering from Cornell University. He then completed postdoctoral training at Massachusetts General Hospital and the Beth Israel the Complex Medical Center at Harvard Medical University with a focus on bone and musculoskeletal research. And he has been an assistant professor since 2020 at Rush Medical College in Chicago with appointment in the Department of Anatomy and Cell Biology and orthopedic surgery. His research integrates bone regeneration, the bone implant intercept, and advanced imaging approach with a particularly emphasis on applying light sheet microscope and the tissue clearing technique to musculoskeletal research. So tissue clearing and light sheet microscope are powerful experiment approach. However, it's always challenging to treat the bone, as you know. And in this seminar, Dr. Ko will discuss several tissue clearing and the imaging technique with particularly focus on the approach on musculoskeletal tissue. Today's title is a tissue clearing and the light sheet microscope imaging of musculoskeletal research. And please join me in welcoming Dr. Franco. Thank you so much.
Well, thank you, Koji. I appreciate the introduction and also want to thank the ISBM ECI for inviting me. It's an honor to present my findings here at the webinar. And also, I actually gave part of this presentation in 2022 ISBM meeting, maybe the first half, I would say. Just putting this presentation together, it was kind of nice to see the progress that we made here at Rush the past few years. So I'm very excited to present our research to the ISBN community. And if you have any questions, feel free to jump in anytime, or if you'd like to save it later, that's also fine too. So I guess we'll get going. So today, as Koji has alluded, the title is the Clearing Enable Light Sheet Microscopy Imaging of the Musculoskeletal Tissues. And specifically, we're going to be looking at bone and the mouse knee. So I wanted to just present some agenda for today. I think separate into kind of three different parts. I want to give some introduction on tissue clearing of the musculoskeletal tissues, followed by our approach on how we're trying to do like sheet microscopy imaging of the knee joint actually, and finally, and the validation study of, is this imaging modality sufficient to detect like biological changes? In this particular case was the intratracheal injection of the nerve growth factor. So let me give you some introductions on the tissue clearing of musculoskeletal tissue. So what is tissue clearing? So essentially, you're trying to match the refractive index of the imaging medium. So the goal is to make the tissue transparent. Normally, if you put a, if you have a certain object, and if you put that object in front of the light, that object blocks the light. And here's an example of here, there's a light that's coming in, that's blocked either by through reflection or absorptions or scattering, and it does not penetrate throughout the tissue because the tissue is opaque, just like your hand or your pencil or your phone or anything. However, the matching of the refractive index actually allows this incident light to penetrate all the way through the tissue of interest. So this image actually was taken from Milton Biotech, by the way. So here's the transparent tissue, and when the tissue is transparent, the light is now able to penetrate throughout whatever the tissue they're interested in, which then allows a lot of different opportunities. I say this, I think I should say this up front, is that light sheet is great, but when the tissue is transparent, you actually could use other imaging modalities such as confocal microscope or any kind of spinning disk, anything that allows that, the focal point to be at different areas of that tissue. Because now you have an opportunity to go deeper into the tissue with the tissue being transparent. Here's an example of some tissue clear sample in the mouse brain. On the left, the figure A shows the mouse brain that's not been cleared. But the B and C are some of the different orientation of the mouse brain that's been cleared by, I believe it was ethylcinamate. And you can see that now, before this grid pattern was not visible, obviously through the scattering or reflection of the light. But now with the tissue being clear, now you can see the grids at the back of this at this particular mouse brain. So that's the goal you're trying to achieve. It's a very simple concept. It's just that when it comes to musculoskeletal tissue, it can be quite tricky. But that tissue clearing effort is also not very recent. In some cases, it's actually been documented in the, I believe in 1920s, if I remember correctly. But just to give you some background, the making the tissue clear, I would say the skeletal prep that a lot of embryologists do is kind of a tissue clearing process. I mean, it's not truly tissue clearing in a sense because you're not matching the refractive index of the imaging media, but you are kind of making the tissue somewhat clear, quote unquote clear. But on the right is actually the actual human hand. I believe that's currently being exhibited somewhere in Germany. And this particular specimen was clear to visualize the blood vessels inside the human bone skeleton. So you can see this zoomed in image showing these different blood vessel structure inside this human hand. So the tissue clearing concept itself isn't very, like very, very recent. It's just that this idea has exploded for the past, I would say, past maybe 10 or 20 years. And here's the PubMed indexed. I probably should update this, probably more since 2024, and because I think I've restricted my search until 2025. But anyway, here's on the right is the results of the PubMed search of, if you put a keyword of tissue clearing since 1912, and you can see this explosion of this tissue clearing article for the past, 10 or 20 years. And there's this particular inflection point in 1969, and I think part of this inflection point is because it's just I don't think anybody actually has confirmed it, but my takeaway has always been that that's the year I believe was the introduction of the very first laser scanning confocal microscope in 1969. And some smart people probably have figured that, oh, this actually opens a lot of different opportunities where if you make the tissues clear, you can then visualize the tissue not just on the surface, but also deeper into the tissue. And I've always been of the opinion that, oh, maybe that's why there's an explosion of this tissue clearing article. And what comes with that explosion of this tissue clearing article is that there's a whole variety of different kind of tissue clearing methods. And they can be generally categorized into two different kinds of clearing methods. One is the organic solvent-based method. The other is the aqueous solution-based method. So one is hydrophobic. So Disco, for example, you may have heard of it. Hydrophilic methods are typically the clarity and cubic. They have advantages and disadvantages of each of these tissue clearing methods. So this is again kind of categorizing into these two categories. So the hydrophobic based method is, or the organic solvent based method is this particular column right here. Oops, sorry. The aqueous-based solution method is here, these three different columns right here. And it has this advantages and disadvantages of each of these methods. And some of the ones that I want to highlight is that the chemicals that are being used, some methods are very hazardous. So organic solvent-based methods tends to be very hazardous compared to some of the hydrophilic-based one, but depends on which hydrophilic-based one, like the hydrogel, can be very neurotoxic. So depends on which method that you go with. And also simplicity-wise, organic solvent-based tends to be fairly simple. It can be very sophisticated for the hydrophilic ones, and clearing performance can be, mixed bag depending on which methods they use. So, it really shows, based on the explosion of this article, shows the variety of techniques that are available for tissue clearing. And when I first embarked on this back in 2018 or 2019, when I first joined Rush, and I talked to my boss, Dr. Sumner, hey, we should do tissue clearing of the bone. And he's like, okay, let's do it, I guess. And I think it was kind of unknown at the time, but I knew there was a paper published like back in 2017 or 16, if I remember correctly, called Bone Clarity. And I didn't know very much at the time, but looking back, maybe this shouldn't have been the first thing that I should have tried. But anyway, so bone clarity is part of the tissue clearing methods in the hydrophilic based ones. And it has this very complicated process. So I would consider this to be hydrogel embedding. So you can see that, it involves a hazardous chemical. Simplicity wise, very sophisticated. So it's kind of a not a good choice for myself because I've never done tissue clearing at the time. And I decided I would choose the most sophisticated one. And plus, it was very slow. And despite the tissue clearing performance to be very high, I don't know, it wasn't very good. Anyway, so this was published in back in 2017 or 16 at the time when I saw this paper as a poster, I thought, this is great, I don't have to do histology anymore. I can just do this tissue clearing and light sheet microscopy all the time and visualize the fluorescent reporter mice that I had at the time. So this is amazing, and the images seem pretty good. And oh, these are SOX9 positive cells, and they seem to be responding to sclerostin antibody treatment. This is all great. But again, this was published in 2017, and it's been about 10 years, kind of crazy, but it's been about 10 years, and there's not a lot of widespread adoption of this particular paper. And this was published in a really great journal like Science Translational Medicine. And again, I think the lack of widespread adoption of this particular technique has been attributed to the sophisticated nature of this technique. I remember trying to replicate this process, like involving nitrogen gas, trying to create this hydrogel, cross-linking, and then also have to be very careful, and then all the process took very long. And as a if you're a grad student or a postdoc, sometimes you're just trying to get stuff done like soon. You don't have time to wait things to work. So it was a very frustrating process. So I could imagine. I mean, I'm not really good at science, but I could imagine other people, even though they're very good, I could see a frustration with this particular technique. So then that's at a time when I started to look into this. Yeah, maybe I should try this organic solvent-based hydrophobic technique. I mean, it seems pretty bad for chemical wise, but at least it's simple. It seems fast, and the clearing performance seems to be pretty good. And at the same time, there was this paper that was published called Pegasus clearing. And this particular paper, I believe, was published in 2018 or 2019, if I remember correctly. And it wasn't, I mean, it was published in the, not like Science Translational Medicine and the other paper, but one of the publishers under Nature. Called Cell Research. But anyway, when it got published, I thought, oh, this is actually quite good. You can actually, this is another paper that shows the ability to do tissue clearing in the mouse, and it's very, very simple. What I mean by simple is that I don't think I have the flow chart here. But compared to what it was shown in the clarity, it's basically a series of solvent solution changes, and then after about a week, then you're ready to image. Whereas the clarity would take 2 or 3 weeks, or sometimes even longer, depending on how you process the sample. So you can see in this published paper different tissues that are being cleared, chest cages, hind legs, kidney, or whatnot, and all the tissues seems to show good clarity, optical clearing, as evidenced by these grid patterns that are easily visible on these images. So I applied this for my own research where at the time, not just at the time, still looking at it, where I'm interested in examining periostin expressing cells during bone regeneration. So these periostin expressing cells are tagged with the TD tomato fluorescent reporter. So essentially what I was trying to do was to visualize these TD tomato positive cells in the bone marrow cavity that are rapidly regenerating. So the next figure shows the mouse femur. The distal end is on the left, proximal end is on the right. So that's where the knee would be. The gray color is the autofluorescence. So just to give you a contextual cue of how the bone femur looks like, and inside this femur are these red positive cells, indicating that all these red positive cells are the periostin expressing cells. And this particular femur was also underwent this procedure called bone marrow ablation surgery, essentially a way to regenerate the bone marrow that fills up the marrow cavity with the regenerating bone. So what's highlighting is that what I'm trying to emphasize is that during the regeneration process inside the medullary cavity, there's a lot of periostin expressing cells labeled by TD tomato that is detectable by light sheet microscopy and tissue clearing. And with that, we can then, rather than simply cutting sections and trying to quantitate the number of TD tomato positive cells, instead of that, we can actually export this data into iMeras, do the automated TD tomato positive cell quantification, normalized by the medullary area, and then boom, we get some ideas of how in the intact, in the intact marrow cavity, there's no periostin expressing cells, but after bone marrow ablation surgery, during bone regeneration, there's the increase in TD tomato positive cells. So that was great, but at the same time, I wasn't quite happy with it because Pegasus tends to give a lot of autofluorescence, surprisingly, and also it was around the time a collaborative opportunity arose with the folks at Rush, who had a very strong interest in imaging nerves in the mouse knee. So that's when I start to kind of shift away from using Pegasus, which I've used in the past, and use something else in the future. But these two folks, Dr. Malfay and Dr. Miller, are actually the main drivers of this particular project. They've always been interested in the neuroplasticity of the mouse knee. I'm not really a OA person, but it was kind of an opportunity for me to kind of develop this particular technique to visualize the 3D nerve innervations around the mouse knee joint. So Dr. Malfay and Miller have always been interested in how the nerve changes around the mouse knee, but again, these images were taken in the 2D sections, and we thought, is there a way to visualize this in 3D? So The problem with Pegasus, as I had alluded earlier, is that there's a lot of autofluorescence. So in the femur, maybe it's okay because perhaps you can chop away as much muscle as possible and then focus on this medullary cavity, but then there's a lot of still extra tissues here. When we first attempted Pegasus clearing, again, it was very not satisfying at all. Here's an attempt to do tissue clearing of the mouse knee joint, and we had to use the lookup table and lower the exposure time to at least try to get rid of the muscles at the outer surface. But this didn't lead to a comparable nerve detection at the knee joint that we would expect to see on the histological sections. So that's when we decided to try different kinds of tissue clearing protocols. So these are all, I would say, organic solvent-based protocols. So the Pegasus here, Sumic here, Disco here is some sort of organic solvent such as butanol, isopropanol, methanol involves varying degrees of harsh solvents. But what's also different from Pegasus or the original Pegasus that I did previously was that it involves the immunolabeling step. So what that means is that instead of relying on the endogenous fluorescence signals, such as TD tomato or GFP or YFP or MTMG, whatever the favorite fluorescent reporter that you're using, we have decided to amplify whether those reporters or any other cell types that we're interested in, kind of I would say 3D immunofluorescence staining, or whole tissue immunostaining. And we tested these three different tissue clearing strategies, and we found out was that here is a mouse knee tissue pre-clearing, and after it undergoes different kinds of tissue clearing process, we found out that the disco seems to be the best in terms of optical clearance performance. And the way we determined that was basically not just visualizing the light sheet scope, as I've shown previously, but also just looking at these grid patterns where Sumic doesn't really clear the tissues as well, like ethylcinemate wasn't doing very much. BB-PEG used in Pegasus also doesn't seem to be, I mean, it is still showing the grid pattern at the back, but we could still see quite a lot of outlines of this tissue and likely from a lot of significant tissue shrinkage. Whereas in the disco, I mean, there's a tissue shrinkage in the disco protocol too, but at least the tissue seems to be quite transparent. I mean, you can see I don't know if you can see the outline, but you can see where the tissue is based on this yellow tint at the middle of this particular image. So after visualizing this particular, or after confirming that perhaps Disco is the way to go for visualizing the mouse knee, so not just femur, but the entire mouse knee joint, it's composed of cartilage, bone, collagen from muscle. So we decided that, okay, maybe for immunolabeling, we should try a different treatment, such as, just like the antigen retrieval that we use for immunohistochemistry. Maybe we should also treat the tissue with collagenase so that it can help the penetration of the antibody deep inside the tissue, because we're worried that in the intact knee joint, it's not going to be able to allow antibody penetration into deep inside the knee joint cavity. So we included those steps and then decided to image the mouse knee. So so the next agenda is just making sure that the light sheet microscopy imaging works. After tissue is cleared, can we label the nerves that we have initially sought out for this particular imaging? So there are different ways to image the clear tissues, as I had alluded earlier, with a confocal, two-photon, light sheet, or some kind of fancy light sheet. And confocal is actually fine, but I personally was not really that was personally not my first choice. for two reasons. One is that it causes the out of plane photobleaching, meaning that here's the confocal light that's coming from the top and focusing on this particular plane, but it also still excites the photons that's out of plane of this particular focal regions, meaning that any of the photons that are being excited here is eventually going to be starved, and we're not going to be able to, perhaps we may not be able to visualize the signals at these outer plane regions as we're changing this focal plane. And the other reason is a lot of the existing confocal microscope, their working distance tends to be pretty limited. We're looking at a mouse tissue that is in the orders of millimeter or sometimes centimeter, I think, depending on how you're dissected. Whereas the confocal scope, I think their working distance, according to Nikon, can go up to 500 microns, but never really had good luck with that. I think 100 micron is a generally pretty good confocal microscope working distance. So it's pretty limited compared to millimeter or centimeter scale that you see in the light sheet microscope. So here is the light sheet microscope. One of the advantages is that as it's shining this laser beam throughout the specimens, it's only exciting the photons at this particular path, not in the path that's out of plane. So meaning that we can, all these fluorophores in the outer plane regions are preserved and they're not going to be bleached by the lasers. So the general setup is kind of looks like this, except instead of brain, we would have a mouse knee here. So the illumination optics would be on the left and actually on the right on the both sides. And as the laser is passing through the sample, anything that's being excited by this laser is going to be detected by the objective on this camera right here. So and this particular scope, Light Sheet 7, is the microscope that we're currently using for all the light sheet experiments. And uh, and and what's the advantage of another advantage, or not just another advantage, but one of the unique thing about this light sheet microscope is that you can move this light sheet up and down, meaning that you can visualize in this, for example, in this brain sample, you can visualize this particular section on here, but then you can shift this light sheet, let's say 100 microns to the top, and then also, again, visualize this particular area at that distance away from the original location. So it's kind of similar to sectioning, but instead of the destructive microtome sectioning, we're doing this optical sectioning throughout the entire samples. So just to give you a little bit of anatomy, so here's the one of the planar image of the light sheet microscope of the mouse knee. The green is the synovial capsules, and blue is the mouse femur, red is the tibia, yellow is the meniscus, and purple is the cruciate ligaments. And you can now see this mouse knee that's been imaged with the light sheet microscope, or cleared by disco and then imaged with the light sheet microscope. And just to give you a look, let me see if I can stop this. Let me see if I can highlight here. So this structure right here would be where the femur is. It's a little bit hard to tell, but trust me, I've seen this plenty of times. And the structure right here would be the tibia here, and this little gap here is where the joint space would be. And that's the autofluorescence signal that's detected by, I believe, was in the GFP channel. And all these red fiber-like structure, string-like structure, or nerve-like structure, is actually the nerves that's innervating inside this mouse knee. And we've labeled this mouse knee with the PGP 9.5, which is one of the pan-neuronal markers. And here is the kind of the projection images as you're going through this entire mouse slice. So like I said earlier, it's like the optical sectioning. You are sectioning through this entire mouse knee joint, and as you're sectioning through the mouse knee joint, you can see these nerves, shows up, disappears, and then goes back. It still has some issues, meaning that the working distance can be a bit tricky. So as you go deeper into this tissue, you'll see that the tissue becomes a bit, opaque. It's an unfortunate thing. I mean, it's a little bit hard to overcome. despite our best effort to clear the tissues, but at least in the area that we're interested in, we can still visualize and detect these nerves in a pretty reliable manner. Um, uh, and whenever we do experiments, it's always important to run controls. So, it's a basic thing. And here on the top is the images that are taken of the sample that's been labeled with just the IgG antibody. So anytime you want to do immunofluorescence, you want to make sure you run this IgG antibody, or at a minimum with no antibody at all. But at least in our case, we ran the IgG antibody, and also on the bottom here is the one sample ran with the PGP 9.5 antibody. And within each respective group, the composite images at the very top and the Alexa Fluor 647. So this is the fluorophore that we use to detect the nerves and the PGP 9.5 antibody staining, but at least we were using the same exact setting to see if there's any signals that light up. So on either in the zoomed images and the 2D sections, we don't see it, but in the PGP 9.5, we see a very robust signaling on the lateral side of the nerve or lateral side of the synovium of this knee joint. And what's also interesting is as we're imaging in, we wanted to confirm that are we actually labeling nerves or are we labeling the blood vessels? So we felt pretty confident that we're not labeling the blood vessels, because the issue with the blood vessels is that sometimes it contains not sometimes, blood vessel contains a lot of red blood cells, which is a heme group, and that tends to autofluorescence quite a bit under a GFP channel, and then also sometimes RFP channel. And we want to make sure that the signals that we detect is not detecting blood vessels. And here is a kind of a zoomed in image of showing that these empty tubular structure are the actual blood vessels. And these nerves are actually some of the nerves are actually traveling along the blood vessels, which is, what we've known for quite some time, but it's actually pretty cool to see in this particular image like here. And also the way we prepared the samples was that we perfused the animals prior to our tissue collections to ensure that we had minimal, minimal blood into the tissues. And here's just a lateral side view of the mouse knee, just highlighting that, yeah, we're detecting a good amount of nerves on the lateral side, indicating that this PGP 9.5 labeling works quite well for detecting these nerves. So that's great. But then the next question is, okay, you can image the nerves and you can image, I guess, those fancy structures, but is this methodology sufficient to detect like biological changes, that alters the nerve structure? And there are a lot of different ways to alter the nerve structure, I guess. Again, I'm not really a OA expert, but according to my colleagues, surgeries or chemical injections or something. But our initial approach was to inject with this growth factor called nerve growth factor, because Dr. Obeida, which was also another collaborator involved in the study, have previously shown that the nerve growth factor causes rapid change in the nerve structure around the knee joint. So we decided that, let's just make sure our method works with this intra-articular injection of nerve growth factor. So here's this background on how we designed the study. Had male mice about 12 weeks old, gave this intra-articular injection of the nerve growth factor twice a week for 4 weeks. And we had 3 groups: uninjected group, saline group, and nerve growth factor group. So kind of 2 controls, uninjected and saline would be the 2 controls. And then nerve growth factor would be the experimental control. And what we found out was that when we imaged the whole knee, so here is the image of the uninjected entire knee. So this is the nerve structure of the entire knee of the uninjected, saline, and nerve growth factor. And when we quantitated this, we actually didn't detect any changes. And when I saw this, maybe this is not very good. But our collaborator told me that you should take a look at closer into the changes where it actually is supposed to change, which is on the medial side of the knee. So this whole knee, the nerve volume percentage is actually for the entire knee. So we decide to focus on the medial side, which is what Dr. Obeda has found that nerve growth factor changes the nerve density. So so we then focused on that particular area where the medial side is. So here's the whole knee with the outline showing the femur and the tibia and where the anterior and the medial side is. And the reason some of the or the uninjected is kind of flipped is because it's on the left knee instead of right knee, like the saline and NGF. And and when we've just focused on the medial side of the knee, so again isolating the mid joint, focusing on the medial side, what we then found out was, oh, there's actually a dramatic upregulation of this rest signal, indicating that there's a high expression of the PGP 9.5 positive signal. So again, uninjected doesn't really show anything. Saline doesn't really show. I mean, there's a little bit, but not a whole lot. But it's really the NGF group that shows this robust, innervation of this PGP 9.5 signals in the medial side of the knee. And here's some images showing that. So uninjected, lateral side is here, medial side is here. Let me see if I can stop it. So that's the lateral side, and then the medial side is going to come forward. And there's not a whole lot here, right? So that's the medial side. And there's not a lot of nerve density on the medial side of the uninjected. I'm going to stop this. And then for the saline, again, this medial side is not really showing anything compared to the lateral side. Lateral side always shows a lot of nerves, regardless of injections. But on the medial side, not all shows up on the saline group. But in the NGF injected group, there's this dramatic sprouting of the nerve on the medial side of the knee joint, indicating that the NGF injection is stimulating the nerve growth, and perhaps it'll allow us to actually help us to determine if this clearing and the light sheet microscopy is sufficient to detect the changes in the nerve. So Spencer, who has a very strong background in engineering, actually has developed this imaging analysis pipeline where through Imaris, he has segmented that medial synovium separately, create this particular volume, and isolated the signals by nerve signals by performing a filtering. So with any kind of immunolabeling, what you'll likely encounter is these remnant dots, either from primary antibody or secondary antibodies. And these dot structures can potentially be not so great when it comes to quantitating the nerve changes. So what he decided was, based on the shape and the size of the structure, he can filter out any of those non-specific signals to isolate these nerves. And following this processing, we then quantitated the nerve density of that medial side and then the lateral side. So on the medial side here, you'll see that uninjected shows a significantly less nerve percentage compared to the nerve growth factor, whereas on the lateral side, all the groups seems to have a similar nerve growth factor. Highlighting that this intra-articular injection of the nerve growth factor promotes the sprouting on the medial side, and also that is easily detectable by this clearing-based light sheet microscopy approaches. So our study demonstrates that I think we are probably maybe the first to demonstrate the imaging of PGP 9.5 positive signals around the mouse knee joints. And to be fair, other studies have also shown, like, for example, Ali Ertug group in Switzerland. Is he in Switzerland? No, I think he's in Germany. Never mind. I think they've shown the clearing and imaging of the whole mouse knee joints, but they've never really examined for like knee innervation in a very rigorous and thorough manner. So I believe we're first to try this. And what really opens up is that we've restricted ourselves to PGP 9.5 antibodies, and restricted ourselves to the knee joint. But I think this particular technique actually opens up other opportunities of using different antibodies. So if you're interested in, I don't know, other types of nerves, like nociceptors or proprioceptors, or not just nerves, but if you're interested in the immune cells or if you're interested in other bone cells, I think by just modifying well, kind of a modular approach, instead of using PGP-9.5 antibody, use a different antibody, Ostrix for osteoprogenitors, or alpha SMA for perivascular cells or something. I think it really opens up different opportunities for different people of visualizing different types of cells that you're interested in this particular tissue. So that concludes my presentation. Thank you for your attention. And this is all these are all the people and cores and the funding sources that contribute to this project. And I'd be happy to take any questions. Mohammed?
Hey, Frank, lovely presentation. Thank you. I was just curious about the size of the images. How big are the sizes of the images? And after you finish imaging them, is it the workflow for quantification is kind of manual or like automatic? Because I know sometimes nerves are tricky in their quantification. So did you set any kind of automatic workflow, or do you have someone who does this for you? Thank you.
So I'm guessing when you refer to the size, like how big file sizes are after imaging,
Yes,
like
I'm sorry. Yeah,
gigabyte
how big
space,
are
right?
the
Yeah,
files?
yeah,
Yeah.
yeah, 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 yeah. That's always a concern, actually. Really depends on how big the samples are, but and how many tiles and how many channels, that all plays a role. But I would say general between 15 gigabytes to, I don't know, 40 gigabytes. I mean, it really varies. Sometimes, I mean, we've had samples that are like four tiles with four channels, and that can go up to 120 gigabytes. But that's very rare because we don't try to push it. But typically for two channels with maybe two tiles, 30 gigabytes, 40 gigabytes, but that's always a huge thing. Like, how are we going to handle this images? And that kind of goes to your next question, like, how do you analyze these? So right now, I would say it's mostly manual. And what I mean by manual is that um, we would export whatever the file to Imeris file, which is the proprietary software that we use. After exporting to the Imeris file, then we would do the filtering, and that filtering is quote unquote automatic. You just have to make sure that the parameter that you use is always consistent with across your all samples. So after the filtering, and then there comes the manual part of drawing the boundaries and then interpolating those boundaries. And then after those boundaries, then we let the Imeris quantitate, okay, this is the thresholded nerves, and what percentage of the thresholded nerve is within that particular boundary that we set out to. So it's a mix of both manual and manual automating, and I wish there's an easier way to do it, but we haven't really found it. But I know like some of the folks in University of Michigan, they're trying to use an AI system to, um, you know, automatically identify like bone features or the, synovial features or something like that. But, I haven't gone to see how much of progress that they've made.
I have a question, Dr. Frank.
Yes.
So actually, I am working on bone and basically on bone cell like osteocyte, which is the most dominant cell and most predominant cell, like 95% cells of bone. And they also have like canaliculi structures, which are very similar to the nerve and all the growth factor. So they can change during, the different types of insults or different physiological or pathological conditions. And I am trying to investigate the changes in the canaliculi network of the osteocytes. So do you have any chance, have you ever, cleared the bone cortical tissues because these osteocytes mainly resides in the cortical compartment and they are very tough. They are very, very heavily mineralized with calcium phosphate and hydroxyapatite,
Right.
those things. So do you have any idea, and can you please guide me through that? How can I get the best of these?
So short answer is don't use light sheet for canaliculi. The reason for that is the resolution is just not good enough. I mean, canaliculi, it's a very thin, narrow canal, and oftentimes you need, I guess you can see a bit of a canaliculi structure for regular H&E, but I've also seen a lot of people use SEM to visualize those canaliculi structures, or those high resolution micro CT to get at least a lacunae and a bit of canaliculi networks. So, but like synchrotron, for example. The reason why I say I don't recommend to use light sheet is the resolution is just not sufficient. We're looking at a light sheet thickness of between 3 microns to 10 microns, depending on your parameters. So that is just not sufficient. Even I don't think it's quite sufficient to detect lacunae reliably. And it's also it's my personal gripes is that when you get these light sheet images and you reconstruct them in Imerus or whatever the favorite software that you use, you can see that the voxels are not isotropic, they're anisotropic. So you have the X and Y's are the equal, the rectangle or square, sorry, of, I don't know, what 0.9 by 0.9 microns. But the Z thickness would be about 2 microns, which leads to kind of a bit of interesting, weird artifacts sometimes as you're going through these Z stacks. So I think there's a lot of work needs to be done. I know there is a paper that came out that they managed to develop this isotropic voxel light sheet microscope. It's just they made it literally out of their backyard, not out of backyard at their institution, but they bought parts from Thorlab and then constructed their own light sheet microscope. So I think there's some technical challenges if you were to try to use this for osteocyte canaliculi network.
Thank you for that question. Yeah, I see somebody raise a hand. Johannes Group. Did you want to ask
Hi.
a question? Yeah.
Yes, hi. Thank you for the great presentation. Maybe in regards to the osteocyte canaliculi, I know of a group that has established some form of confocal imaging for that in mineralized samples. I'm not sure if you're aware of that, but
No, I'm not aware of it.
but yeah, there's some.
Yeah, I have
think-
seen that. I am also familiarized with that. But yes, I was thinking to have some idea from this light sheet microscopy. But people have done some confocal microscopy and they have shown the canaliculi network. Yeah.
Yeah, confocal would be probably better for that particular question, I would say.
Mhm. Mhm.
My question would be, maybe I missed it in the beginning, but which of the forms of tissue clearing would you now recommend the most for bone in regards to both applicability and yeah, I guess the results?
So let's say you're just doing femur. Let's say you're just doing femur tibia. I think and you want the least amount of trouble, and you have a mouse that has fluorescent reporter. Pegasus might be the best because it's the simplest and it's easiest and it's quickest. But let's say you don't have a bone that's not, that doesn't have endogenous fluorophores. In that case, you'll probably have to use some sort of a disco-based protocol. So using immunolabeling to target the cells that you're interested in. And but then, the thing is, this tissue clearing is always evolving. I've seen there's actually a paper from Sandra Scheffelbein's group who used the Life Canvas system to visualize the cortical bone structure of a mouse. They've also incorporated the in situ hybridization. So I think that might be a pretty good approach as well. It's just that I don't think they've used that for like other complex organs like mouse knee, for example. They've demonstrated pretty convincingly in the mouse cortical bone, but just not in the mouse knee. So in general, for simplicity wise, Pegasus might be a way to go. But then, if you want more adaptability, disco, I would push to the disco side with the different immunolabels. And then I would say live canvas might be a kind of a wild card. The other issue with live canvas is that you have to buy their equipment and their own reagents, which can be pretty expensive. So it's another caveat.
Okay, thanks.
Koji?
Yep. First of all, thank you so much for the wonderful, like beautiful imaging and beautiful lecture, educational, and it's definitely like a helpful for the people who want to start or like having a trouble in light seed microscope and tissue clearance. So I never performed the like tissue clearance, but like let me ask you a very basic one. Like I feel like if we perform the long decalcification procedure, that can also like affect the
Yep.
antibody bindings. But like based on your, some of the previous try, I didn't see like any like a different condition in terms of the decalcification. I'm wondering like did you try a little bit longer or shorter, like a different method for like maintain the beautiful, like for maintain the antibody bindings? Like, do you have any insight about that part?
So I think that's a very valid concern. I mean, that's, sounds like you have a very good experience with immunohistochemistry in general and probably troubleshooted different kind of decalcification. So all those concerns actually can be applied for what I presented today, especially when it comes to labeling. I mean, I even mentioned a little bit antigen retrieval steps too. So um, in my personal experience, I've personally have never encountered any issues whether I use EDTA or formic acid. But formic acid, like 20% formic acid, but that doesn't mean that it's going to work with every kind of antibodies, right? Because some antibodies work well with the formic acid decal, some antibodies work well with the EDTA, but with a certain percentage of EDTA, right? Are you using 14% EDTA or 20% EDTA with a pH of 8 or
Right.
pH is, it's a whole different world, right? And then you have to this whole different, parameters of what kind of antigen retrieval are you using? Are you using collagenase? Are you using hot citrate or using the proteinase K, I mean, you probably understand this, right? So I think that's a valid concern. So one thing that, if you don't want to this whole ordeal of tissue clearing prior, you may want to validate on like the tissue sections first to make sure that it's actually working. So if you get like a paraffin sections or cryo sections, just go through similar steps and make sure that whatever the decal or even fixation, for example. I mean, I've seen studies where even 4% PFA and 10% formalin yields a different immunolabeling too, right? So, so you may want to do a quick test, like validation step test on the tissue sections to make sure it works, and then move on to the tissue clearing. That way, you're not wasting the entire like tissue. Because once there's tissue cleared and imaged, you're kind of done. I guess you can reverse clear it and then repurpose for histology, but the morphology is not great. So it's something of a step that you can take before embarking on this whole journey.
Perfect. Thank you so much.
There's a question in the chat from Kim Dang Kyun. Thanks for your presentation. They're working on a dental implant model and wanted to ask if you have any idea for tissue clearing and visualize collagen fiber and soft tissues around epithelium surrounding implant in titanium.
I thought I saw a paper on, oh my God, on bone research. I think the group was using, um, oh my goodness, it's one of the Pegasus developer. I mean, he's not the main driver, but he's included as a co-author of this paper where they inserted the titanium implant into a mouse that are expressing leptoreceptor creatine tomato, and tried to visualize that into the mandible. But I could also be just looking at the bioarchive, not the bone research. But I know people have done it, and that paper, if you try to look for that paper, it might be the one that help you in your project.
All right. Any other questions? I can
Great.
ask one. Yeah, I
Yep.
can ask one question. Is that you said compared to the aqueous protocol, this hydrophobic protocols is pretty quicker or faster. How
Mhm.
long does it take to clear the knee joint?
So if you were to include a decalcification, it can take 2 to 3 weeks.
Okay.
Depending on how fast, I mean, there's like formic acid can take 3 days, right? But EDTA can take up to 2 weeks. So it really depends of what you decide to do. Like Pegasus, for example, if I remember correctly, including with the decal, sometimes you can do it within a week. But Disco tends to take longer because there's the immunolabeling steps of primary and secondary antibodies. So it's something to consider.
Are there any considerations regarding concentration of antibodies that you used for this compared to like
Um,
what we do in 2D sectioning?
Yeah, so I generally use lower concentration than maybe 2 or 3 folds lower than the general like immunohistochemistry. Let's say you're using, I don't know, on top of my head, like 1 to 100 or 1 to 200 dilutions for IC, so immunofluorescence. Sometimes 1 to 500, it could be 1 to 1000, but I generally go with 1 to 500.
Okay, good to know. Mohammed, you had a question?
I'm sorry, this conversation is so nice. So, with the antibodies, do you do like a passive antibody staining, or do you use because I know sometimes with the bigger volume or the bigger tissue, the penetration of antibody when you do it passively doesn't penetrate very
Yeah,
well. So,
yeah.
do you use any techniques to ensure that there is a good penetration?
Yes. I'm glad you pointed. I'm glad you asked the question. So, this is actually inspired by one of my colleagues in Denmark, Thomas Henderson, who used vacuum systems to kind of exchange the solutions inside and out. So essentially, you have a desiccator with the sample sitting inside, and then that desiccator is hooked up to the vacuum pump, and that vacuum pump turns on and off every hour or two, and that changes in the pressure helps the solutions go in and out of the tissue. So that seems to do the trick in sometimes, but you know, probably depends on how the antibodies made too. Some antibodies I think are much bigger than the other antibodies, so we'll see. I know they're like nanobodies too. Um, but the problem is I mean, I don't know if anybody has good lucks with them. Like for nanobodies, I've had good lucks on like the tissue sections, but every time I tried on like the entire tissue for tissue clearing, it just never really works. So um, I don't know, it's just kind of mystery to me, those nanobodies.
So another question came into my mind now. I know that one of the very essential things for the clearing and techniques is a good perfusion. Like you need to have a good perfusion since the
Mhm.
beginning. So do you just perfuse with PBS and formalin, or do you use, I know some people use heparin or something
Yeah.
earlier to ensure the clearing of blood cells. So do you do something special?
No, we just use PBS and formalin, but yeah, I've heard about people using or inject the mice with heparin first and then use a PBS mixed with heparin for perfusions. But in our case, we've just used PBS followed by formalin, and that seems to do the trick.
Great. All right. We have another minute if somebody wants to ask a question. All right. Thank you, Dr. Ko, for this great
You.
talk. Yeah. And thank you all for showing up. We have such a huge crowd joining us today to learn about light sheet and tissue clearing. I guess everybody is interested in trying this technique out. Just a reminder again that our next seminar is on writing abstracts for the upcoming ISBM conference. And you can find the registration link in the chat. So please join us next time. And thank you, everybody.
Thank you. Bye.